how it is going to affect the system in the cell, and how the changes are taking place in the human being. That is what we are going to see. So before we will start with the actual translation transcription process, we need to understand the basics of nucleic acid and nucleotides. So in today's lecture, we are going to see the nucleic acid and nucleotides along with nucleosides. So this is the syllabus already we have discussed yesterday. Now I will start with the nucleic acid and nucleotides. So there are two types of nucleic acid, you know, that is deoxyribonucleic acid and ribonucleic acid. The primary nucleic acid serve as a repository and transmitter of genetic information. So repository means what all the basic inherent characters are there in your father, in your parents, in your mother, that will transmit to you. Okay, it is a repository. Repository, they are storing it and they are transmitting. The storage and transmission of the particular information related to your parents will be done by DNA and RNA. So we see the functions of nucleic acid. So DNA is the chemical basis of hereditary and may be regarded as a reserve bank for genetic information. So obviously it is a chemically it is taking place, and what all the information I told you already, hereditary information, transformation from one generation to another, second to third, third to fourth. So it is like a reserve bank which is giving you the particular genes from your ancestors, your grandparents, to your parents, to you and your children. Like the next is the DNA is exclusively responsible for maintaining the identity of different species of organisms over million of years. So suppose different species, suppose in any race or in any particular family, if suppose the father height is above 6 feet, so obviously the daughter or son height will be around 6 feet. If the father is having a cat eyes, obviously the daughter or son will be having the cat eyes. These are the chances are there. So this particular thing happens with respect to disease also. If father is having some disease, the child may be having, or the son or daughter may be having that particular chances of having that particular disease. Okay. And this is what the identity of different species they are responsible. This is a particular identity that particular family is having, and the transmission of that particular identity from one generation to another generation. Further, every aspect of cellular function is under control of DNA. So any small function, whatever is there inside the cell, is under the control of DNA. Now we'll see the components of nucleic acid. So nucleic acid are the polymers of nucleotides. It is also called as it is a polymer. It is also called as polynucleotides held by 3 days and 5 days phosphate bridges. You know very well this fact. 3 days and 5 days phosphate bridges. I told you yesterday also, if possible, please revise your 11th, 12th or 8th, 9th, 10th science book so that at least you will get a basic idea with respect to this. And also I request, if possible, please sit with pen and paper so that what all the structure, what all the basic thing and some short forms that I am going to show you, which is useful for you to remember throughout your life, that you please note it down immediately. In other words, nucleic acid are built up by monomeric units, that is nucleotides. Okay, small unit of nucleotide is made up of. It may be recalled that protein is a polymer of amino acid. So we know that protein is protein. For both protein is a small single monomer units of amino acid. Now we'll see the main thing, nucleotides. Now what is nucleotides? TIDES. Nucleotides are composed of nitrogen bases, then pentose sugar, and a phosphate group. Nitrogen bases are adenine, guanine, cytosine, thymine. These are the different nitrogen bases are there. Then a pentose sugar, that is ribose, and a phosphate group, PO4 minus. So this phosphate group, if these three things will combine together, then the structure will become a nucleotides. Okay. So nucleotide perform a wide variety of functions in the living cell. Besides being the building block or monomeric unit in the nucleic acid. So as I told you that small function in the cell will be done by DNA, and all the DNA are made up of nucleotides and nucleosides. So it is indirectly going to control all the components of cell. Next terminology is nucleosides. The term nucleosides refer to base plus sugar. So in the previous case we have seen that nucleotide means, if you see this particular slide, base plus sugar plus phosphate, and we are see the next terminology nucleosides. Nucleosides are base plus sugar, that is nucleoside. Now these are the two different terminology, and I saw the student, good student, ranker student, those who are, when they are appearing for the exam, they are making a small mistake and they are writing here. In the case of nucleoside, they are writing it is composed of base plus sugar plus phosphate, and as a result they are getting less mark. The answer is wrong. Now there is something you need to remember, some tricky thing you need to remember, that the difference in between nucleotide is made up of base plus sugar plus phosphate, and nucleoside is made up of base and sugar. Now, how to remember this? Okay. So, there is one simple thing technique I wish all of you should write down. This, now you can see over here in the slide how to remember this particular. So, first point that we have seen is base plus sugar plus phosphate group. So, this particular thing is tide, TIDE. We have highlighted with red color. Next is base plus sugar is nucleoside, SID. So second point you can see side is base plus S, sugar, and tide is base plus sugar plus phosphate group. So you can see over here where the cursor is there. Now the final line is S side of S is BS, and the first letter of tide T is EST. So here there is a simple and small trick to remember is BS here and ES here is ES. Write down this point, note on this, and take a photo of this slide. And remember, if you connect this particular information out your life, today I am reiterating and repeating the same thing. Nucleoside, SID, S indicate ES, base, sugar. When if you know this tide means base and sugar, then obviously for tide, that is nucleotide, you can write down it is made up of base plus sugar plus phosphate group. I hope I am clear. If you have any doubt in the next lecture, we are going to take this online lecture next time. So during that particular period, I will discuss this. I request anyone or Gusti to note down this point. If any doubt is there, you can consider or you can take during the online lecture. So the slide is made up of base, sugar, and base. Next, now structure of nucleotides. So this nucleotide is composed of purines and pyrimidines. Now these purines and pyrimidines are the nitrogenous bases which is found in the nucleotide, and therefore it is nucleic acid. We have the aromatic hydrocyclic compound. Now purine structure and pyrimidine structure, if you will see, we have the aromatic, that is six membered compound, cyclic structure, and heterocyclic. Now why it is called as heterocyclic? So you can see over here where my cursor is there, this slide, this structure, where you can see that it is a six-membered cyclic structure. So it is aromatic. In the previous slide, we told that aromatic is aromatic. bases are of two types, purines and pyrimidines. So structure of nitrogen base we are going to see. Why request follow please know the structure. Side by side I will explain you. Purines are number purines are numbered in the anti-clockwise direction while pyrimidines are numbered in clockwise direction. So those who are preparing for GPAT exam, GPAT, those who want to pursue their M pharmacy in India, that it is very much important. Over here, you can see that purine structure is given. So in the purine structure, if you see, these are the international. This numbering was given by International Union of Pure and Applied Chemistry (IUPAC) system. So 1, here 2, then 3, 4, 5, 6, 7, 8, and 9. So you see that numbering was given was in anti-clockwise direction. While pyrimidine was given numbering clockwise 1, 2, 3, 4, 5, and 6. So these are the basic difference in between the numbering system of purines and pyrimidines. I hope all of you do this structure, and also request to write down the numbering system and remember the numbering and explain that why it is so assigned. So the according to the international system, it is given. So it is well defined system that we are directly adopting. Next one is sugars of nucleic acid. Now we have seen the bases, that is nitrogen bases. Now it also contains sugar, that is made up of base plus sugar. So the next point is sugar. Now 5 carbon monosaccharide, that is pentoses, are found in the nucleic acid structure. Now if you see the basic structure, 5 carbon monosaccharide. So if you see over here, 1, 2, 3, 4, and 5. 1, 2, 3, 4, and 5 carbon. Now these are the 5 carbon monosaccharide, single unit of glucose or sucrose or saccharide, that is pentoses, are found in the nucleic acid structure. Now RNA contains D-ribose while DNA contains deoxyribose. So D-ribose and deoxyribose. Okay. Now here the main difference in between the RNA structure and DNA structure is ribose and deoxyribose differs in structure at E2. You can see over here 1, 2, and here also 1 and 2. So here you can see that deoxyribose, one oxygen less at C2 compared to ribose. Here the one oxygen, you can see my cursor over here. Here the one oxygen is less than that of the ribose. So this RNA contains hydroxyl group, whereas DNA contains only hydrogen group. Now this is the one of the distinguishing point in between DNA structure and RNA structure. And because of this presence of hydroxyl group over here, this RNA undergoes several other chemical reactions that we are going to see today only. Okay, now all of you clear? I hope all of you drew this structure. Please take your time, pause this slide if you want, as it is a recorded lecture. I wish all of you to pause this particular point, draw the structure correctly, and again revise the previous structure, that is purine pyrimidine structure and ribose and deoxyribose structure. Now here what happens when this particular ribose is attached with the base. This is the base. When this particular ribose is attached with the base, here also numbering is given 1, 2, 3, 4, and here also the numbering is given 1, 2, 3, 4. So there is confusion, chances of creation of confusion is there. So to avoid this confusion, when this sugar is attached with the base, this sugar is numbered 1 dash, 2 dash, 3 dash, 4 dash, and 5 dash, like that. That we will see in the next slide, but I am just keeping you posted. So these are the distinguishing characters in between the numbering of base over here and the next is sugars. Okay. So till time we have completed the nucleotide. What is nucleotide? What is nucleoside? Composition of nucleotide is base plus sugar plus phosphate. Composition of nucleoside is base plus sugar. Now these bases are of two types: purines and pyrimidines. Purines and pyrimidines. This is the structure of purine. Purine is numbered anti-clockwise, while pyrimidine is numbered clockwise. So these are the main distinguishing characters. Then after we have completed the sugars, then we have seen the D-ribose sugar and D-deoxyribose sugar. The main distinguishing character in between is Sp2. In the DNA, there is presence of only hydrogen, whereas in the D-ribose, that is in the RNA, there is a presence of hydroxyl group. So till time it is clear, and I hope you are understanding it. If not, I again request you to note down the points so that in the next coming one or two lectures we can discuss on that particular point when I will take online lecture. Then nomenclature of nucleotides. The addition of pentose sugar to base produces nucleoside. So in the base and sugar is nucleoside. I told you. If the sugar is ribose, the ribonucleosides are formed. Adenosine, guanosine, cytidine, and uridine are the ribonucleosides of A, G, C, U respectively. So if you see the bases are adenosine, guanosine, cytidine, and uridine are the nucleosides of A, G, C, and U respectively. If the sugar is deoxyribose, deoxyribonucleosides are produced. So this is the nomenclature. Now another thing is the term mononucleotide is used when single phosphate moiety is used to a nucleoside. The term mononucleotide is used when a single phosphate moiety is added to a nucleoside. So mono indicates one, a nucleotide which contains base plus sugar plus phosphate, then it is called as mononucleotide. A single phosphate moiety is added. Thus adenosine monophosphate, AMP, adenosine monophosphate contains adenine, ribose, and phosphate group. So I hope this is clear. What the full form of adenosine? Adenosine monophosphate. Adenosine obviously SIN sign S contains sugar, that is adenine. This adenosine contains two parts, adenine and ribose, and monophosphate itself indicates mono means one and phosphate means phosphate group. So adenosine, ribose, phosphate, one group is there. So collectively it is called as AMP. Now, there, as I told you that in the previous slide, there are adenosine, guanosine, cytidine, and uridine. These are the different bases are there. Now, depending upon that, if we will add sugar, the nomenclature will change. So, principal base, nucleoside, and nucleotide. Principal base is adenine, guanine, cytosine, and uracil. So, we add nucleoside, that is sugar over here, then it becomes adenosine, guanosine, cytidine, and uridine. And if I will add phosphate group in it, then it becomes adenosine iodide monophosphate, or it is also called as adenine. And it is abbreviated as AMP. Like guanine is the base, guanosine is a sugar added component, and guanosine iodide monophosphate or guanylate is a phosphate group added component. So here the base, here the sugar, and here the phosphate. So it becomes a nucleotide. The abbreviated as GO. The same is the case with the deoxyribonucleotide, where adenine, deoxyadenosine is sugar added component, and deoxyadenosine iodide monophosphate or deoxyadenylate is a phosphate group added component, and it is abbreviated as DAMP. So I hope all of you clear with this particular base, its sugar component and its phosphate component, and how it is abbreviated. The same is the case with the ribonucleosides. It is base, sugar component, and this one is the last one is the phosphate group component. Now next one is the binding of nucleotide component. Now I told you that a nucleotide is composed of base plus sugar plus phosphate. Now these all three components are joining or combining with each other, but at which place and how the particular structure will form. I request all of you, please note down this point very carefully. The atoms in the purine ring are numbered to 1 to 9 and for pyrimidine as 1 to 6. We have seen the numbering system. If you see your notebook also, if you drew the structure, you can see, you can cross-check. That is, purine is numbered 1 to 9. Purine is anti-clockwise, while as pyrimidine is clockwise. So please remember this. The next point is the carbon of sugars are represented with an associated prime dash for differentiation. I told you in the previous slide only that as and when the sugars are attached with the base, sugars, whatever the numbers I told you in the previous slide here, this is the one number, but when it combines with the base, pyrimidine, the sugar will be numbered as 1 dash for easy differentiation. And it is mentioned over here prime dash for differentiation. Thus, the pentose carbon are 1 dash and 1 dash to 5 dash because it is pentose sugar having 5 carbon. So, we are writing it not as 1 to 3, but we are writing it 1 dash, 2 dash, 3 dash. The structures of two selected nucleotides, namely AMP and ATMP, are depicted here. So, now we see over here. One contains adenosine monophosphate. Adenosine monophosphate is a purine base. Now we are going to see that adenosine structure numbering will start from here 1, 2, 3, 4, 5, 6, 7, 8, 9. You can see the ninth position. Here the ribose sugar is attached. You can see the numbering which is over here is 1 dash, 2 dash, 3 dash, 4 dash and 5 dash. You can see over here. So here it is a base, here it is a sugar, and at fifth position there is an attachment of phosphate group CO3 minus. Okay, so this is a particular one structure for adenosine monophosphate. So this is a adenosine. Adenosine contains two things. That is S. S means base plus. This is a base plus sugar. So this contains adenosine and ribose. And then at which position it is attached? So attachment of sugar to the base is at ninth position of base and first position of sugar, whereas phosphate group is attached directly at fifth position of sugar. So this is the structure of adenosine 5' monophosphate AMP and thymidine 5' monophosphate. The second structure is thymidine monophosphate. So thymidine is a pyrimidine analog or pyrimidine derivative, whereby the numbering is starting from this anti-clockwise 1, 2, 3, 4, 5 and 6. Okay, so now this thymidine is attached with the ribose sugar. The same is the case over here. The remaining structure is very much same as because monophosphate is there here and thymidine which contains the ribose sugar. Now if you want to attach a second phosphate or third phosphate group over here, you can attach over here, the stick mark is given. So it becomes diphosphate or triphosphate. Adenosine diphosphate and adenosine triphosphate the structure will become like this. So I wish or I request all of you to draw the structure of AMP and TMP, thymidine monophosphate. I'm again repeating, adenine is a base, ribose is a sugar, and phosphate is the phosphate group attached. It is monophosphate, so it becomes adenosine monophosphate. Second structure is thymidine monophosphate. Thymidine is a pyrimidine, thymidine monophosphate. So this is the ribose sugar. Phosphate. Please in your notebook very carefully along with the numbering. Again, when if possible, revise the structure. If any doubt, please note down it. We'll discuss. Okay. Now, structure of DNA. So, till time we have completed adenosine, then nucleotide is composed of base plus sugar plus phosphate, whereas nucleoside is composed of base plus sugar. Then we have seen the structure of purine and pyrimidine. will go ahead with the structure of DNA. So now that some theoretical part will start. DNA is polymer of deoxyribonucleotide or simply it is deoxynucleotides. It is composed of monomeric units, namely deoxyadenylate, deoxyguanylate, deoxythioidylate, and deoxythioidylate. These are the main four components are there: deoxyadenylate, deoxyguanylate, deoxythioidylate, and deoxythioidylate. It may be noted that some authors prefer TNT for deoxythioidylate since it is found in DNA. Now it depends upon the author to author if some variation is there, but whatever the there in the learning year and Satyanarayana, that is what we are going to cover in our lecture. Now schematic representation of polynucleotides. Now this is a very important structure and figure that you need to remember. Now how the structure of DNA is formed, and how the monomeric units are combining with each other, and how the longest structure of DNA formed. That is what we are going to see. So on the right hand side you can see there is a structure is given. So right now we have seen the single structure of adenosine monophosphate. Now this adenosine monophosphate structure, how it becomes, how it form a bond with another structure, and how the elongation of the chain is taking place, that we are going to see now. Okay, the monomeric deoxynucleotides in DNA are held together by 3-5' phosphodiester bridge. Now you can see over here phosphodiester bond. So ester means ROR. So this is ROR, and here also it is ROR. So these are the diester bonds whereby the phosphate bond is held together by 3-5'. Now 3-5' is written because 3 dash of 1 dash, 2 dash, 3 dash. All of you can see my cursor. 1 dash, 2 dash, and 3 dash. 3 dash of guanine is attached with the 5 dash of thymidine. So that's why it is called as 3-5' diester bridges. First thing. Second, DNA or RNA structure is often represented in a shorthand form. So shorthand form is also given over here. The horizontal line indicates the carbon chain of sugar with base attached at C1. The horizontal line indicates carbon chain of the structure. So here the carbon chain, the horizontal line you can see, and where the base attached at C1. So here at one position the base is attached. Now near the middle of the horizontal line is C3 dash phosphate link, while at the other end line C5 is the phosphate link. You can see instead of this we have C over here. The horizontal line indicates carbon chain of structure with base attached at C1. Now you can see over here in this particular thing, C1, adenine is a base which is attached at C1 of sugar. Sugar with base attached at C1 dash. So this C1 dash where the adenine is attached. Adenine is the base and this is the sugar. Near the middle of the horizontal line is C3, the phosphate link, while at the other end of the line C5. I told you this C3, 1 dash, 2 dash, 3 dash. This C3 is attached with the C5 by phosphate bond, that is phosphodiester bond. I'm repeating again, the DNA are held together by 3-5' phosphodiester bond. That is one unit is attached to another unit by phosphate group. That phosphate group is attached at 3 dash of one component and 5 dash of another component. Then second thing is the base is attached at C1. So this is adenine is base and it is attached at C1 of the sugar. And this 3 days is attached with the 5 days with phosphate. This is what all about the structure. Next thing, DNA double helix structure. So double helical structure of DNA was proposed by Lance Patterson and Francis Crick in 1953. And for that, several scientists were awarded with Nobel Prize in DNA free double helix model. DNA structure is considered as a milestone in the era of modern biology. But after the 1960, there is evolution in the era and the particular field of biology and genetics. And the structure of DNA double helix is comparable as a twisted ladder. So this particular structure you might be knowing, and it is a double helix structure where both the strands are running parallel to each other, and it is a 3 days, 5 days structure running towards a central axis. You can see this is a central axis. Okay. So we'll see in detail about this structure. So first point is the DNA is right-handed double helix. DNA is a right-handed double helix. It is a right-handed double helix structure. It consists of two polydeoxyribonucleotide chain. It consists of polydeoxyribonucleotide chain twisted around each other on a common axis. It is twisted chain on a common axis. You can see my cursor over here. This is the common axis on which it is particularly twisted. The two strands are anti-parallel. That is, one strand runs in the 5 days to 3 days direction, while the other in the 3 days to 5 days. This is a 5. You can see the color black and red. So it is a 5 days, and you can see the 3 days. Here it is a 3 days. It is a 5 days. This two, sorry, this is comparable to two parallel adjacent roads carrying traffic opposite direction. Next, we see the inner structure with the distance, how the distance is there. So the width or diameter of the double helix is 20 angstrom. You can see over here, it is the diameter which is, it is 20 angstrom or 2 nanometer. Each turn pitch of the helix is 34 angstrom or 3.4 nanometers. Each turn, one turn, which is a 3.4 nanometer with 10 pairs of nucleotides. So you see there are 10 pairs of nucleotides placed at a distance of about 0.34 nanometer. So each pair is having a defined distance, that is 0.34 nanometer. So the diameter is around 2 nanometer, and each turn, that is helix, is having 3.4 nanometer, and each nucleotide pair is placed at 0.34 nanometer. 3.4, 0.34, and 2 nanometer. These are the 2 to 3 things you need to remember. Okay. Next, each strand of DNA has hydrophilic deoxyribose phosphate backbone. Hydrophilic deoxyribose phosphate backbone, that is 3 days, 5 days phosphodiester bond on the outer periphery of the molecule. Now this is a molecule, this is the outer periphery. Okay, so this outer periphery of the molecule is having hydrophilic deoxyribose phosphate. So S means sugar and phosphate. S sugar, phosphate, sugar, phosphate, sugar. So the outer periphery is covered by sugar and phosphate group. Okay, in both the sides, sugar, phosphate, sugar, phosphate, sugar, phosphate. Okay, so the outer periphery is having sugar, phosphate on the outer side of the molecule, while the hydrophobic bases are stacked inside. So base, it is adenine, thymine, guanine, cytosine. Adenine, thymine, guanine, cytosine. This particular base, which are hydrophobic bases, which are placed or stacked inside the particular structure. I hope I am clear. Please try to understand. I am again repeating. Outside of the structure is sugar, phosphate, which are hydrophilic. Sugar is hydrophilic. Phosphate is hydrophilic, dissolved in the water, like that you will remember. So if it is hydrophilic, it dissolves, it will be outside. And the bases, which are the nitrogenous bases, which is insoluble in water, adenine, thymine, cytosine, guanine, this particular thing are placed inside the structure. Inside the structure, that is core. Okay. And the two nucleotides chain are not identical but complementary to each other. They are not identical but running complementary to each other. continuously due to base pairing. So base pairing is given over here ATCG, ATCG. Now we'll see A bond with T and C bond with G. So the composition of AT and CG with respect to structure now we are going to see. I'm just giving you a rough idea. A bond with T with two double bonds, whereas cytosine and guanine bonding with triple bond. Now how? That is what we are going to see the structure. So you can see that adenine and thymine AT is having a two double bond. So I request all of you to draw this structure, and after this we'll see how bonding is taking place. 1 minute. Now here if you see the two strands are held together by hydrogen bonds formed by complementary base pairs. So base pairing are there. It is adenine is a purine derivative and thymine is pyrimidine. So this is one of the base. This is another base. This base pairing is taking place by two double bonds. Whereas in the A and P pair has a two hydrogen bond, while CG pair has a three hydrogen bond. You can see over here cytosine and guanine. 1, 2, and 3. There are the three. I request all of you to draw the structure and remember the structure or rewrite the structure as and when you get the time. So if you see the strength, so CG is stronger by about 50% than adenosine and thymine. So adenosine and thymine is having two double bonds. Cytosine and guanine is having 1, 2, and 3 double bonds. Now, comparatively, if you see the strength wise, cytosine and guanosine is stronger than that of the adenine and thymine. I hope I am clear. In this particular figure, also previous figure, all the dimension we have seen, that is diameter is 2 nanometer, then each turn is having 3.4 nanometer, and each pair is placed at 0.34 nanometer. Outside of the structure, that is hydrophilic sugar and phosphate is present, and inside the structure there is hydrophobic base AT and EG with two double bond and three double bond respectively is present. Okay. So I think this structure, I request all of you to draw the structure right now. Once the slide is possible, and you can see over here two chain is written, and here also two chain. So this is one of the components. Again, it will be attached with the another chain over here, and again third chain, fourth chain. The sequence is continuous. Okay. Next, the hydrogen bonds are formed between purine and pyrimidine only. If two purines accept each other. Now, for this purpose, important thing, two purines cannot sense the bond. They would not fit into allowable space because two purine cannot fit because one purine. So one purine is equal to whereas pyrimidine is very small space. As a result, they are fitting in the space. And likewise also, two pyrimidines would be too far to form a bond. So the two pyrimidine bonds are very much far to each other, so they cannot form a bond. So in nutshell, the bond formation is taking place in between two different bases, that is purine and pyrimidine only. If purine cannot form a pair because both the things cannot fit into the allowable space. Another thing is pyrimidine cannot form a pair because both the pyrimidine molecules are very far from each other to form a hydrogen bond. So to have a particular bond of double bond or triple bond, there is an utmost need of two different bases, that is purine and pyrimidine. The only base arrangement possible in DNA structure from a special consideration is AT or TK. A can form a bond with C or T can form a bond with A and CG or GC, like this. Okay. So I hope all of you clear with this. Next point is the complementary base pairing in DNA helix proves Chekhov's rule. Now, Chekhov's rule is one of the rules Important rule over here, you need to remember and note down also. Sugar rule. The content of adenine equals to that of the thymine and guanine is equal to that of the hydrogen. In the DNA particularly, if you see that consider that adenine is 5%, 10%, 50%, obviously thymine content would be also 5% or 10%, receiving equal concentration. Okay. So this DNA helix is giving or is proving the Chagav rule, whereas RNA is not following the Chagav. That is what we are going to see when we see the RNA. Okay. So as long as content wise also, if you see adenosine is in equal proportion with thymine and guanine is in equal proportion with hydrogen. Structure of RNA. Now before I will go with the structure of RNA, we'll just take a quick revision. So till time we have seen the this one nucleotide and nucleosides, whereby nucleotide contains, rather we will start with the nucleoside. As I told you, the base contains sugar, nucleoside contains base and sugar, whereas nucleotide contains base, sugar and substrate molecule. Then after we have seen the composition of base, the mainly bases are composed of two things, that is purine and pyrimidine. Purine is a 9-membered compound and pyrimidine is 6-membered compound. Both are aromatic tetracyclic compound. Purine runs in numbering of the purine is anti-clockwise, whereas pyrimidine is in clockwise direction. We have seen the structure of ANC and PNP, adenosine monophosphate and adenosine thymidine monophosphate. Then we have seen the structure of sugar, where the sugar ribose and deoxyribose is there. It's a 5-membered structure where the difference of DNA in the deoxy sugar, there is a presence of only hydrogen at C2, and in the case of ribose sugar, there is a presence of hydroxyl group at C2. Then after we have seen the complete double helix structure of DNA, whereby the numbering system is there, different uh, different diameters are there. For example, the complete width or diameter of double helix is 1 twist or 1 turn. The length is 3.4 nanometer, and each pair, the distance between each pair is 0.34 nanometer, and both the thing is revolving around the central axis, and outside of the particular structure there is a presence of sugar phosphate, that is a hydrophilic component, and inside there is hydrophobic component, that is base. Then after we have seen the structure of adenine, thymine and cytosine and guanine, where we have seen how it is attached by 2 double bonds and 3 double bonds. Okay, I hope I am clear, and if you are having any doubt, we'll discuss later on. Then we have seen the Chagav rule. Chagav rule indicates the content in the DNA. The content of adenine is equal to thymine and cytosine is equal to guanine. So these all are the points we have seen. Structure of RNA is there. We will see some basic points of structures of RNA. This RNA is a polymer of ribonucleotides held together by 3 days, 5 days diester between the same as that of the DNA. Although RNA has a certain similarity with DNA structure, there are specific differences. Like now, we need to see what all the differences are there in between DNA structure and RNA structure. So first is pentose sugar. The sugar in the RNA is ribose in contrast to the deoxyribose in DNA. So if you see, this is deoxyribose sugar and this is ribose sugar. So this ribose sugar is present in RNA first thing. Second, pyrimidine. RNA contains pyrimidine uracil in place of thymine. So instead of 80 adenosine bond with the thiamine in DNA. Here, adenosine bonds with the uracil, ADU. Okay, this is one of the difference. Third is single-stranded. RNA is usually a single-stranded polynucleotide. However, the strand may fall at certain places to give a double-stranded structure. Generally, we have seen that DNA is a double helix structure, and here it is a single-stranded polynucleotide. The first difference is pentose sugar, ribose sugar. Second difference is pyrimidine is forming a bond with uracil instead of thiamine. And third is single-stranded. Fourth, I told you Chagos rule. It is not obeying the Chagos rule because due to single-stranded nature, there is no specific relation between purine and pyrimidine component. So because of the single strand, there is not necessary that if adenine is this much percentage, so pyridine will be this much percentage. So it is not obeying Chagos rule. And second last point is susceptibility to alkali hydrolysis. Alkali can hydrolyze RNA to 2-3' cyclic diester. Now, why it is happening? Alkali cannot hydrolyze DNA, but it is hydrolyzing RNA only. Why? This is possible due to presence of hydroxyl group at C2. So here you can see my cursor is there. At C2, hydroxyl group is there. Here in the DNA, there is only the hydrogen group. So because of the presence of hydroxyl group, it is susceptible to hydrolysis and it is converted into 2-3' cyclic diester. The DNA cannot be subjected to hydrolysis due to lack of this group. And the last one is porcinol color reagent. This porcinol color reagent is one of the experiment is there in the biochemistry where RNA can be histologically identified by porcinol color reaction due to presence of ribose. When the ribose in the presence of porcinol reagent, it will give you a characteristic color. So that is one of the important things with respect to. So there are some points and so there is present pyrimidine is there, that is uracil is there, single stranded, Chagos rule is not obeyed by this, and susceptibility to alkali hydrolysis, and the last one is porcinol color reaction. So up to this we will cover the types of RNA in detail. We'll take in the next lecture. I request all of you to draw the structure, put down the important points. Because I am coding over here, and if you have any specific doubts, I again request you to your Nikita or any other person we believe that discuss with you to find the point and discuss with you so that I will cover it and take the online lecture and offline lecture. Thank you very much.